company here. So yeah, I've tailored this presentation to focus more on serology testing. So I know that I believe that you're using both of our serology assays in the lab. So I thought this would be maybe more interesting and relevant for you. So let's get started. This is the outline of my presentation. Lots of topics that are going to be covered, but my presentation is about, I think, 35 minutes long, so it's lots of small sections. So I'm going to be covering different topics around antigen testing, including, sharing the characteristics of our assays, sharing some data that's been published, both in terms of antibody production after a natural infection, and now we're also interested in looking at antibody production after vaccination, now that the vaccines are being rolled out to a big part of the population. So let's start with a more general introduction about diagnostic testing for SARS-CoV-2. So there are two main categories of tests for SARS-CoV-2 that you are probably aware of. So first, we have the tests that detect the presence of the virus. So these tests will use an upper respiratory sample because the virus is present in the respiratory tract. So either a nasal or a nasopharyngeal swab, which we saw more commonly at the beginning of the pandemic. So the swab that went quite deep into the nasal cavity. And so these are really used to determine if someone is currently infected by the virus and needs to isolate. So in that category, we have the PCR tests, which detect the viral RNA in the sample. And we also have antigen tests, which became more popular recently. And so these antigenic tests will typically detect the nucleocapsid antigen of the virus. And then we also have tests that detect the presence of antibodies that are generated in the patient's blood after infection or after vaccination. And so this will be more the focus of my presentation today. And so these tests can be used to determine if someone was previously infected by the virus as part of a natural exposure, or we can now use them to see if antibodies have been produced and to what extent following vaccination against SARS-CoV-2. So for all of these types of tests, we have either central lab version of the test or point of care versions of these tests. This figure here shows the course of molecular as well as serological biomarkers, both during and after an infection by SARS-CoV-2. So during the acute phase of the infection, the presence of the virus in the respiratory tract, as I mentioned in the previous slide, can be detected by either PCR or antigen testing. So these two markers will typically start to rise a few days before the onset of symptom in the case of a symptomatic infection. And while the detection of the antigen is known to be closely correlated with the presence of infectious virus in the respiratory tract, as shown by the green and orange curves in this figure, the RNA as detected by PCR is known to remain detectable for a longer period of time. And this is because PCR has a much higher analytical sensitivity. And it's also the gold standard for the detection of SARS-CoV-2 infection. And then around 2 weeks after the onset of symptom, we have IgG and IgM antibodies that will start to appear in the blood of the patient. And based on the data that's available so far, circulating antibodies against SARS-CoV-2 remain detectable for at least 8 to 12 months after a natural infection. After vaccination, this is something that's still under investigations as the vaccines have been rolled out earlier this year. And then, so we know that the IgG, we know that the IgM and IgG antibodies typically start to rise at the same time. So IgMs do not appear earlier than IgGs. They basically appear at the same time. And while IgG antibodies will last for a much longer period, in terms of IgM, we see that they remain for about 2 months. They remain detectable for about 2 months before disappearing. So if we take a closer look at the antibodies that are produced after exposure to SARS-CoV-2, we know that some antibodies will be directed against the spike protein, which is found at the surface of the virus. And so this is the protein that is used by the virus to enter host cells. And we also have antibodies that will be generated, which target the nucleocapsid antigen, which are shown in red in this figure of the virus. So the nucleocapsid is present in several copies, and it's basically associated with the viral RNA. So it's found inside of the virus. So these are the two main types of antibodies that are produced after exposure to the virus. And as I've mentioned in the previous slide, in contrast to what is generally observed with other infections, in the case of SARS-CoV-2, IgM and IgGs appear mostly at the same time, and we even see numerous cases where IgGs have actually appeared earlier than IgMs. So in the case of SARS-CoV-2, the presence of IgM is not a specific indicator of early infection. I mean, it indicates early infection, but IgGs are present too and could be used as a marker of early or later infection. We also know that there is a lot of variability that has been observed in the serological profile of patients, both in the timing of antibody production after infection, as well as in the levels that are reached in these patients. And when we look more closely at those antibody titers, what we've seen is that there doesn't seem to be a direct correlation with the clinical severity of the disease in terms of antibody levels that are reached versus clinical severity of the disease. So now I'll provide an overview of the characteristics, and I will go as well into more details in the design that we're using for our two antibody assays for SARS-CoV-2. So Roche has two serology assays for the detection of SARS-CoV-2 antibodies. Both assays are now approved for use by Health Canada. So the first assay that we've launched in the summer last year, so it was launched in the summer of 2020. This assay is meant for the qualitative detection of antibodies that are generated against the nucleocapsid of the virus. So because it targets the nucleocapsid, this assay can really be used to determine if a person was previously exposed to or infected by the virus as part of a natural infection setting. It could also be used to detect a natural infection that would occur after someone has been vaccinated, as long as the vaccine that's been used targets only the spike protein, which is the case with the vaccines that are currently available in Canada. The second assay allows the semi-quantitative detection of antibodies that are targeted against the spike protein of the virus. So like the anti-N assay, this assay can also be used to determine if a person was previously exposed to or infected by the virus. But in addition, because the spike protein is the target of the vaccines that are currently used in Canada, this assay will be able to be used to monitor antibody titers following vaccination. We can also see at the bottom of the figure the clinical performance of these two assays based on our internal data. Sensitivity, which is typically assessed in PCR positive samples at 14 or more days after confirmation by PCR, ranges between 98.8% in the case of the NTS assay to 99.5% with the NTN assay. And in terms of specificity, it's very high. These assays were really designed with the goal of maximizing specificity in order to minimize false positives. And so it ranges between 99.8 to 99.97 for these assays. So our serology assays use recombinant proteins that represent either the nucleocapsid antigen in the case of the NTN assay, or the receptor binding domain of the S protein in the case of the NTS assay. And so for both of these assays, we use a design called the double antigen sandwich, which is sometimes shortened to DAGS. So this is the same design that we use for our blood screening assays. And it actually allows the preferential detection of high affinity antibodies. We know that most high affinity antibodies are also IgGs, and we'll talk more about that in the next few slides. And also, as I mentioned, our goal was really to maximize the specificity of our assays in order to minimize false positives. So here, I just want to take a moment to show you what actually happens in the samples when we put them on the cobas analyzer. So it basically shows how the assay works. So first, the patient sample will be incubated with a mix of biotinylated and routinylated antigens from the virus. So either representing the nucleocapsid antigen or the spike protein of the virus, depending on the assay. And so then we have these immune complexes or double antigen sandwich, so whereby we have the antibody that is attached to two antigen. And so these complexes will be formed, of course, if the corresponding antibodies are present in the patient sample. Then we will add streptavidin-coated microparticles, which are shown in purple here in the figure. And these will bind to the immune complex via the biotin-streptavidin interaction, as one of the two antigens here is labeled with biotin. And then finally, the mixture is transferred to a measuring cell, where the microparticles will magnetically fix to the electrode surface, and luminescence will be induced by applying a voltage to the electrode and measured with a photomultiplier. So that's how we're measuring the signal and the presence of antibodies in the sample. Now, I want to take a step back and go over this slide with you, which basically shows how antibodies develop and mature over time following an infection by SARS-CoV-2. I want to show you this because it will be relevant in the next few slides when I go into more details on how our serology assays are able to specifically detect high affinity antibodies. So I want to show you a little bit, what are high affinity antibodies and where do they come from. So early after the infection, the immune system will typically respond to the presence of the pathogen by initiating a mass production of antibodies. And these antibodies will typically have low affinity for the virus, and they will include IgA, IgMs, and IgGs. So these early antibodies that are produced in response to the infection will not effectively recognize the virus, but since there will be many of them, their low affinity for the virus will be offset by the high number of antibodies that will be produced. Among the antibodies that are produced during the early response phase, a few may arise from immune memory from other coronavirus strain exposure, and show some specificity to SARS-CoV-2. Then over time, the immune system will improve its recognition of the virus by directly interacting with SARS-CoV-2, and it will generate more antibodies that are highly specific to the viral antigens. And so this is called the affinity maturation process, whereby the immune system will generate antibodies of increasing affinity to the target antigen. And then in the late response phase, at that point, the antibody pool will be comprised of very specific high affinity antibodies that are predominantly IgGs. And so as part of this pool of high affinity antibodies, we may also have what we call neutralizing antibodies, which are able to effectively inactivate the virus and prevent its infection. And so the specific detection of these high affinity antibodies is an interesting aspect, because the detection of these high affinity antibodies will be more likely to correlate with neutralizing capacity and potential immunity, as we know that these neutralizing antibodies are a subset of the high affinity antibody pool. So basically, this is what our assay was designed to do. So the Alexis assay was really designed to preferentially detect high affinity antibodies. And so in the next few slides, I want to show you side by side the main difference between two commonly used antibody assay formats. So on the left, we have the double antigen sandwich assay, which is the format that we use with our Alexis assays. And on the right, we have another commonly used antibody assay format, which is the indirect assay format. And so in both cases, we of course need to start with a sample. So that serum or plasma sample may contain different types of antibodies, IgGs, IgMs. Some of them might have low affinity or high affinity to the virus, but this is the starting point. And then both assay formats, of course, must use an antigen as part of their content in order to capture and detect antibodies that are present in the sample. So in the case of the double antigen sandwich assay, the complementary antigens to the antibody and those will be labeled with biotin or ruthenium. And so these complementary antigens will be added to the sample. And a key characteristics is that the antigens will remain in solution in the sample. Whereas in the indirect assay format on the right, in this case, the antigens are attached to a solid substrate. And so this is really the key, the first key difference between these two assay formats. And then in the double antigen sandwich assay format, what happens is that each antibody must independently bind to two antigens in order to be able to generate a signal. And so as the antigens in this case are free floating in the solution, it will require a good strength of binding between the antibody and the antigen. And therefore, these assays will favor the detection of high affinity antibodies because of the fact that it requires a good strength of binding between the antibody and the antigen. In the case of the indirect assay formats, we have high affinity antibodies that will be able to the antigens on the solid substrate, but because the antigens are fixed on a solid substrate, we also have low affinity antibodies that will be able to bind to the antigens because basically there's availability of several binding sites, one next to each other. And so this will allow low affinity interactions to take place between the antibody and the antigen. So this refers to the concept of avidity. And so avidity refers to the combined strength of binding across all binding sites between the antibody and the antigen. And so a high avidity can be generated by several binding sites of low affinity. And so this basically highlights the main difference between the two assay formats. So the double antigen sandwich assay really requires the independent binding of two complementary antigens and will therefore preferentially detect high affinity antibodies. Whereas in the indirect assay format, because the antigens are fixed on a solid substrate, this will allow high avidity binding, and it may generate cross-reactivity with low affinity antibodies. The second key difference between the two assay formats is in the way that the signal is generated. So in the case of the double antigen sandwich assay, we have antigens labeled with ruthenium that are used with this assay, and therefore the signal is directly generated via these bound antigens. And then in the case of indirect assays, we need a second antibody in order to detect the primary antibody and generate a signal. If the secondary antibody is specific to IgG, it will allow these assays to only detect IgGs and specific to IgGs. However, since both high and low affinity antibodies can bind to the antigens on the surface, these assays have an increased risk of detecting cross-reacting antibodies. And so basically, just because an assay is IgG specific doesn't mean that it will be specific for high affinity or neutralizing antibodies, which is the antibody class that is important for conferring immunity. So this slide here shows some of the use cases for the anti-N or anti-S antibody assays. So there are different applications, of course, for the use of antibody tests. We can use either of these assays to look at seroprevalence in the population. These assays, again, both of them could be used to look at antibody levels or the simple presence of antibody in an individual patient, either to see if the person was previously exposed to the virus or if antibodies were produced after vaccination. And in some cases, it could also be used to complement PCR in a diagnostic setting. In the case where we suspect that the individual or the patient was infected by SARS-CoV-2, but the PCR is repeatedly negative, then we could wait for antibodies to be produced and use serology testing as a means to confirm prior infection. We can also use the two assays in conjunction with each other. This could be useful for seroprevalence studies in a low prevalence setting. So the idea in this case would be to perform sequential testing in order to improve the positive predictive value of the test in a low prevalence setting. And it could also be used, we could also use both assays together on the same patient in order to increase sensitivity. There's been a few reports of individuals in which we were detecting only anti-N or anti-S antibodies in these patients. I don't know if it's because one of the two antibody types were just too low to be detected, or if it's really the immune response that was specific to one antigen versus the other. But this could be used as a means to increase sensitivity. And then we also have the anti-S assay, which based on the vaccines that are currently available in Canada, which target the spike protein of the virus. So these antibody assays, of course, can be used to look at the vaccine-induced immune response on at the antibody level. And because the assay from Roche is quantitative, this assay could also be used for the characterization of convalescent plasma for donation. So in the next few slides, I will show you some data that contrasts total versus IgG specific antibody assays. Here, the authors in the study compared the diagnostic sensitivity of several commercial antibody assays, including the Alexis NTN assay. So they basically evaluated a total of 73 blood samples from individuals with mild to moderate disease or asymptomatic infection. Among these samples, 25 were collected 2 to 3 weeks after PCR confirmation and are shown by the green bars. And 48 of these samples were collected more than 4 weeks after PCR confirmation and are shown by the blue bars. As we can see, the specificity of total antibody tests, such as those from Roche and Siemens here, was significantly higher at more than 4 weeks after PCR confirmation than for IgG-specific assays. The authors suggested that this could be due to the ability of total antibody tests to detect all immunoglobulin class. So this is one hypothesis for the increased sensitivity provided by total antibody assays. In this other study, the authors used, again, various commercial antibody assays to explore, in this case, the temporal changes in antibody levels for a period of up to 10 months after COVID-19 in both hospitalized and non-hospitalized symptomatic patients. As we can see again here, the total antibody assays showed higher positivity rates. And they also reached their highest positivity rate sooner than the IgG-specific assays. Again, showing higher sensitivity in the case of total antibody assays versus IgG-specific assays. Here, the authors evaluated antibody levels again over a 10-month period. With either total or IgG-specific antibody assays. So in this case, the study included 201 samples that were collected from 84 patients with a PCR-confirmed SARS-CoV-2 infection. So with the total antibody assays, which are shown in the upper part of the figure, we see that the antibody levels were sustained for the entire study period, whereas with IgG-specific assays, which are shown on the lower portion of the figure, we see that the antibody levels tended to decrease over time. So this is a significant difference in terms of clinical interpretation, because basically, when we use total antibody assays here, the conclusion is that the antibody levels are sustained over time, whereas if we use IgG-specific assays, then the conclusion would be that antibody levels are waning over time. So it is a critical difference in the case of the clinical interpretation in the end. Here, I just wanted to show you some internal data that was generated by Roche. This is specific for our Alexis NTN assay. So we basically looked at the duration of antibody response against SARS-CoV-2 by evaluating sequential donations from individuals with a prior SARS-CoV-2 infection, which was confirmed by PCR. So as I said, the antibodies were measured with our Alexis NTN assays. And we included in this data analysis donors who had at least 5 drawings and a minimum of 50 days of testing. So in the figure on the left, we basically have 10 donors who had more than 100 days of sampling. And in the figure on the right, we evaluated 49 donors with more than 50 days of sampling. And so as we can see in all tested samples, the cutoff index values, which is the result provided by the qualitative Alexis assay, so the values remained either constant or increased over a period of up to 128 days after infection, showing basically that the antibody levels are sustained up to more than 4 months after infection. So now I wanted to show you some data from our quantitative NTS antibody assay, and it's used in the context of vaccination. So around the globe, there are currently more than 50 vaccine candidates for SARS-CoV-2 that are under investigation, and 8 of them have been approved for full use in various countries. In Canada specifically, we have 4 vaccines that have been approved for use by Health Canada, including that of AstraZeneca, Moderna, and Pfizer, as well as the one from Johnson & Johnson. However, the vaccine from Johnson & Johnson, which has the advantage of requiring only one dose, this one has not yet been released for distribution in Canada due to an ongoing investigation into a manufacturing issue. All of the vaccines that are currently used in Canada target the spike protein of the virus, which means antibody assays that target the spike protein, or more specifically, the receptor binding domain of the spike protein, can be used to monitor antibody production after vaccination. So there are already several published studies showing the usability of Elexis antibody assays in the context of vaccination. So the Elexis NTS assay, of course, can be used to evaluate antibody response after immunization with vaccines that specifically target the spike protein, or they could also be used with whole virus vaccines, such as the one from Sinovac. And then the Elexis NTN assay can be used in the context of vaccination to either screen individuals prior to vaccination to assess their antibody status, or to monitor the occurrence of a natural infection that could occur after vaccination. The NTN assay could also be used to evaluate antibody response after vaccination with the whole virus vaccines, like the one from Sinovac. So these whole virus vaccine will elicit an antibody response to both the spike and nucleocapsid, and so therefore they could be used, but this vaccine is not currently approved for use in Canada. I'll now show you some data from a few publications which use the Roche Elexis NTS assay to monitor the production of antibodies after vaccination in healthy individuals. So here, antibody titers were measured over 1 month after the administration of the first dose of the AstraZeneca vaccine in a group of 135 healthcare workers. Among this group, there were 44 subjects who already had antibodies prior to receiving their vaccine. And so these are shown in green, and the remaining 91 subjects were seronegative prior to vaccination. And so these are shown in blue. In the seronegative group, we see that the majority of individuals did not have any measurable antibody response at 7 days after receiving their first dose. Rather, the antibody levels started to increase at 14 days post-reception of the first dose, but there were still 3 individuals which had not developed any measurable antibody response after 28 days in this group. We can also see that the median antibody response at 28 days in the seronegative group was similar to that of seropositive subjects at baseline and was on a rising trajectory. And then in the seropositive group, the antibody levels were 200 to 500-fold higher than that of the seronegative group. Here in this other study, antibody titers were measured over 50 days following the administration of the Pfizer vaccine in a group of 255 health workers who were aged between 25 and 70 years old, as well as a group of 112 elderly subjects aged 80 to 95 years old. So the 2 doses of the Pfizer vaccine were administered on day 1 and 22. We can see that the antibodies started to increase on day 22, which is about 3 weeks after receiving the first dose. Then they reached a plateau on day 36, which was 2 weeks after receiving the second dose, and started to decline on day 15, which was basically a month after receiving the second dose. The antibody levels were inversely correlated with the age of the subjects. And so they were basically lower in older individuals and also tended to decrease a bit more quickly in this group. There's also emerging data on the production of antibodies following vaccination in immunocompromised patients. So here, antibody titers were measured 2 to 3 weeks after administration of the second dose of the Pfizer vaccine in a group of 167 patients with chronic lymphocytic leukemia. So in the figure on the left, we see a comparison between 52 patients with leukemia and 52 age and sex-matched healthy controls. And we can see that only 52% of the leukemia patients developed antibodies, whereas all of the individuals in the control group did develop antibodies after vaccination. In the figure on the right, we see that among patients with leukemia, the antibody response rate varied between 16 to 79%, and this was dependent on the treatment or disease status at the time of vaccination. There's also a publication that reports the case of a patient with multiple sclerosis who was under treatment with ocrelizumab at the time of vaccination. So ocrelizumab is a monoclonal anti-CD20 antibody which will deplete both B and T cells in these patients. And so the patient still had not developed a measurable antibody response 27 days after receiving his second dose of the Pfizer vaccine. And so based on these preliminary results, it seems that antibody response following vaccination is markedly impaired in immunocompromised patients, and it is also affected by disease activity and treatment at the time of vaccination. I want to take a moment to talk a little bit about neutralizing antibodies. So these antibodies are a subset of antibodies that can effectively prevent the infection of host cells by the virus and are a key indicator of immune protection. So we know that more than 90% of antibodies against SARS-CoV-2 are directed against the receptor binding domain of the spike protein. So these antibodies are able to inhibit the attachment of the virus to host cells by blocking access to the ACE2 receptor, which is used by the virus to enter host cells. So the commercial immunoassays like the Roche Elexis assays are able to detect and quantify antibodies that specifically bind to the receptor binding domain of the spike protein, some of which may be neutralizing. However, these assays are not meant to directly measure neutralizing activity. In order to measure neutralizing activity, we really need to use specialized assays that are meant for that purpose. And so far, the minimum antibody titer that is required for protection against SARS-CoV-2 infection or the development of severe disease has not yet been established. Here we examine the correlation between the results obtained with the Elexis NTS assay and the plate reduction neutralization test, which is basically the gold standard method for the assessment of neutralizing capacity in a blood sample. So we used a total of 390 plasma samples from convalescent donors who were analyzed in parallel with both methods. And so from the data that we see here, basically when using the Elexis assay cutoff of 0.8 unit per ml, we obtain a very high positive agreement of 99.4% and a positive predictive value of 98.9% between these two assays. Now, if we push our analysis further and if we set the Elexys assay cutoff at a higher level, so at 15 unit per ml, in order to optimize the agreement with virus neutralizing capacity, we obtain in this case a positive percent agreement of 92.5%, but the positive predictive value is increased to 100% at that cutoff. So the Elexys NTS assay, based on this data, we see that it correlates very well with the detection of neutralizing antibodies, and it could be used as a surrogate marker for the presence of neutralizing antibodies in a sample, for example, as part of the characterization of convalescent plasma for COVID-19. And there are already guidelines around this in terms of the level of antibodies that should be used for convalescent plasma donation. In December 2020, the World Health Organization released the first international standard for anti-SARS-CoV-2 antibodies. Here we evaluated the correlation between the units of our Elexys NTS assay to the units of the international standard using pooled plasma from 11 individuals that had recovered from a SARS-CoV-2 infection. As we can see in this figure, we have an almost perfect linear relationship between the two scales with a Pearson correlation coefficient of 0.9996. So basically the conversion of the Elexys units to the international standard units results in really minimal numeric changes that are well within the range of typical assay variants, and therefore the application of a conversion factor is not required, and the Elexys units can be considered equivalent to the units of this new international standard. This is something, this data will be included in the next version of our package insert for this assay. Now, this is the last portion of my presentation where I basically want to provide a brief update regarding the use of antibody assays here in Canada and elsewhere. Right now, there are currently 22 serology tests for COVID-19 that are authorized by Health Canada. So these include both lab-based and point-of-care antibody tests. So basically, there are many choices of assays that are currently available on the market for antibody testing. These are the guidelines from the Ontario Ministry of Health for the clinical use of antibody testing for SARS-CoV-2. So currently, antibody testing is only allowed for specific clinical indications in Ontario, including the testing of patients with multisystem inflammatory syndrome or patients with severe illness who have tested negative for COVID-19 by PCR, and where antibody testing may help inform the clinical management of these patients or public health action. And so at this time, antibody testing is not allowed to determine immune or vaccination status in Ontario, but this is something that might change, especially in the case of immunocompromised patients. I was made aware that currently in BC, antibody testing is done on myeloma patients because we know that these patients have a poor antibody response following vaccination, and these patients are being offered a third dose of the vaccine in order to try to enhance their antibody response. You may have heard about the ongoing collaboration between Roche and Moderna. So Moderna is using our Elexys NTS assay as part of their clinical trials to quantitatively measure antibody levels following vaccination with their vaccine. And so this is done in order to help establish a correlation between vaccine-induced protection and the levels of NTS antibodies that are generated after vaccination. And they are also using our Elexis NTN assay to assess seropositivity at baseline in their trial participants. So there are still many unanswered questions when it comes to SARS-CoV-2. A lot of them are around the topic of immunity. So for a long time, since the beginning of the pandemic, we've wondered if antibodies confer reliable immunity, and if so, for how long they would confer this immunity. It's still unclear at this point what is the contribution of cellular immunity. So we talk a lot about the humoral or antibody levels, because this is easier to measure. Cellular immunity is something that's a bit harder to measure, but there are ongoing investigations on that aspect of immunity as well. And then I think one of the key questions right now, it is the most burning one as we're gaining more data around antibody production, is really what is that minimum antibody titer that would be required to confer protection against COVID-19. So this is an area of ongoing investigation, and hopefully we'll have more answers soon. So this completes my presentation. I see we have a few minutes left, and I will be happy to take any questions you might have at this point.
If anyone has any questions, you can just come off mute, or you can place it into the chat and I can share it with everyone.
Meeting. I think there was something in the chat. I would ask if I'd be willing to share the slides. And yes, I'll be happy to do so. I just need to get them approved. And as soon as they're approved for distribution, I'll be happy to share them with you, Dr. Tian.
I should also mention that I have recorded this presentation, and I'll be sharing it with Jennifer to share with anybody who'd like to see it again, or for anybody that missed it. It'll take a little bit of time for Zoom to create the recording link, but once I have that, I will share that.
Great, thank you. That was a great session. I'm sorry I missed part of it because I had a conflict. I think it was very clear, and it helps to answer some questions as well. So thank you very much. And I think we will learn a lot more. So from the clinical perspective, I think antibody testing may be similar to hepatitis B in the sense that what we are seeing is that there are some people who don't develop antibodies. We actually have had patients that we call clinically diagnosed with COVID. And so, but the antibodies are negative, thanks to sort of Roche support of the reagents. We were able to test this population that were not lab confirmed. And some of these patients, when we give them vaccine, they're still negative a month out. And so I think we will, like moving forward, what I see from the clinical end is going to be like, may not be mass testing of antibodies per se. In certain groups, I think there will be testing to make sure, like healthcare workers, right? Mm-hmm. Like if it's 2 out of 100, like even thinking about UHN and Sinai, there are a lot of people that may not be mounting antibodies despite that they are vaccinated with 2 doses.
Mm-hmm. Yes, especially those immunocompromised patients. But I think this relates a bit to what is being done with hepatitis B testing. They don't, of course, they don't test everybody after vaccination to make sure they have antibodies. I mean, in healthy people, this is less of a concern, but in healthcare workers who may get exposure in immunocompromised patients, this makes more sense. And then yes, with hepatitis B also we have a cutoff established, so under which people are advised to get boosters. So eventually we might get there with COVID-19, hopefully. But thanks for showing your clinical insight, Dr. Chiong. I'm more on the I look at the articles and everything, but it's always great to know what's going on in the real life with patients. One of the questions that is asked on the chat is, do we have studies showing the correlate of protection, basically at what level we are considered immune? And so this is the burning question. We don't know yet that magic number above which there would be enough antibodies to say, okay, you're protected against a future infection. We have some insights, as I've shared in one of my slides, which is maybe I can show it here. This one here, we know that the presence of neutralizing antibodies is good correlate of immune protection, because these are really the effective antibodies. And at a cutoff of 15 units per ml with the Roche assay, we see that all of the samples who were above that level also had neutralizing antibodies detected with the gold standard virus neutralization test. I mean, is this enough or not enough? I mean, these are the type of experiments that are hard to run, because I guess over time we would need to look at patients with certain antibody levels to see whether they are reinfected at that level or not. So I think we're getting there. We're also getting more insights as to the levels that are reached after vaccination, what you could expect as a patient. So yeah, this is that famous level of, or that famous antibody threshold is not yet established, but I think eventually we'll get there and have those answers. So I also got some kind words about the presentation. So thank you so much. I really appreciate your nice comments. It's always nice to hear. And I don't know if there were any remaining questions at this point. I mean, we're almost at the end of the hour, so I'm happy to take any last minute question if there are any.
Caroline, it's Jennifer from Mount Sinai. So I don't have a question. I just wanted to say thanks for tailoring this presentation today. We have 4 research groups on the line as well as the core lab. So I think everybody learned a lot for sure. And there's still so much more to learn about the antibody response. So many questions about looking at antibodies with different viral strains or T and B cell responses, but I really liked a lot of the data that you included with recent publications. So thank you for that. I know that I get questions on a weekly basis about COVID serology, but definitely if I ever come across something that I need your help with, I'll let you know as well.
Yes, please feel free to connect directly with me. I'm guessing the type of questions you might get, and I'd be happy to reach out to my colleague in Europe to see, what's being done there, or see, from my contacts, the more we exchange, the more we find out. So it's always a strength to share knowledge. And so thank you so much for your kind words. It was my pleasure to do this presentation to your group this morning, and I appreciate your time. Great.
Thank you very much, Carolyn, for providing this presentation this morning. And just to keep everybody on time and back to their day, it's just approaching 11 o'clock. So we wanted to thank you very much for joining our presentation today and providing an opportunity for Caroline to share this presentation. So thank you, Caroline, and thank you everyone for joining.
Thank you all. Enjoy the rest of your day.
Thank you. Bye for now.
Bye.